Good afternoon, ladies and gentlemen. My name is Eric James Meehan, and I'm with my colleagues today, Joe Mitchell, Helena McShea, and Sharifa Al-Shabri. Today, we are here to talk to you about 3D bioprinting. Three-D bioprinting technology shows potential in tissue engineering for the fabrication of scaffolds, cells, tissues, and organs reproducibly and with high accuracy. Bioprinting technologies are mainly divided into three categories: inkjet-based bioprinting, pressure-assisted bioprinting, and laser-assisted bioprinting. I will now pass you on to my colleague Sharifa, where she will give you a brief history of 3D bioprinting.
Hello everybody. Today in this video, a brief history of bioprinting will be mentioned. The promise of printing human organs began in 1983, when Charles Hall invented stereolithography. The type of printing relied on a laser to solidify a polymer material extruded from a nozzle. The instructions for the design came from an engineer who would define the 3D shape of an object in computer-aided design software, and then send the file to the printer. Hall and his colleagues developed the file format known as STL. It carried information about the object's surface geometry represented as a set of triangular faces. By the early 1990s, 3D systems had begun to introduce the next generation of materials, such as nanocomposites, blended plastics, and powdered metals. These materials were more durable, which meant they could produce strong, sturdy objects that could function as finished products. By 1999, Wake Forest Institute of Regenerative Medicine used a 3D printer to build a synthetic scaffold of a human bladder. And in 2003, Thomas Boland was able to modify an inkjet printer so it can accommodate and dispense cells and scaffolds. Also, the first bioprinting lab was opened, where bioprinting was practiced regularly. A year later, a technology that can engineer tissues with only cells was developed. Other major points in the history of bioprinting was 2007, where the first bioprinting company was founded, and 2009, where the first commercial bioprinter, NovoGen MMX, was introduced. Thank you very much for listening.
Hello everyone, my name is Helena McShea. Today I will be discussing some of the biomaterials that are used in three-dimensional bioprinting. According to the Online Medical Dictionary, a biomaterial can be defined as any substance other than a drug, synthetic or natural, that can be used as a system or a part of a system to treat, augment or replace any tissue or organ or function of the body, especially materials which are suitable for use in prosthesis that will be in contact with living tissue. When we are selecting a biomaterial to use, the material properties, implant site requirements, and manufacturing technology should all be taken into consideration. The biomaterials used must be printable in nature, meaning they possess printability characteristics, such as rheological properties, biocompatibility, such as no cytotoxicity or immunogenicity, and the possession of appropriate structural and biomechanical properties. I will be talking about the main biomaterials that are used to bioprint cells, tissues, and organs. The various technologies used in 3D printing are chosen according to their intended function. Biomaterials that are used to 3D bioprint can be classified into metals, ceramics, polymers, and various combinations of these, and living cells or tissues. Metals that are used are metal powders. Some examples are titanium powder, stainless steel powder, tantalum, inconel, nickel-based alloys, and titanium aluminides. Ceramics that can be used are for medical modeling. The most common ceramic used is gypsum. Some other examples are zirconia, alumina, and hydroxyapatite. Polymers can be used in 3D printing to form bone implants and grafts. They can also be used to form scaffolds for replacements of bones. So these are some of the various biomaterials that are used in 3D bioprinting. I'm going to hand you over to my colleague Joe, and he's going to discuss some of the challenges faced in 3D bioprinting.
Bada bing! Hi guys, my name is Joseph Mitchell, and today I'm going to be talking about some of the challenges and limitations facing the 3D bioprinting industry under three headings: costs, regulations, and technologies. When addressing the cost of bioprinting, I'm going to talk about some of the pioneers of the industry. Organovo are a company that has the technology to bioprint smooth muscle, blood vessels, and endothelial cells. Organovo's revenue doubled from 2010 to 2012 to 1.2 million, although in the same year they had an operating loss of over $9 million. Tevido is a company based in Texas, and they develop breast tissue aimed for cancer survivors. This company believes that they will need at least 40 million over the next 7 years to continue to develop their products. $15 million expected to come from US federal grants, with 25 million must be gathered from venture capitalists. Unfortunately, since the sector is so young, revenues so low, and returns so slow for investors, there is not many willing to put their money forward for this technology. With regards to regulation, currently there is no regulatory framework in place specifically for 3D bioprinting. As the development of this sector is happening at such an alarming rate, legal framework has been lagging behind innovation. Current legislation has classified bioprinting as an advanced therapy and is regulated under the Advanced Therapy Medicinal Product Framework by the European Commission. Although there is a lot of scope for advancement in application, bioink technologies still need to be further developed before we can expect to see a fully bioprinted organ. Despite these challenges, there is a lot of hope and expectations for the 3D bioprinting industry, and my colleague Eric is going to bring you through some of the advantages in today's world and prospects for tomorrow's. Thank you.
Thank you, Joe. I'm now going to talk to you about the advances in today's world from 3D bioprinting and an indication of where it will take us in the future. Organs. While an entire organ has yet to be successfully printed for practical surgical use, scientists and researchers have successfully printed kidney cells, sheets of cardiac tissue that beat like a real heart, and the foundations of a human liver. Although the production of an entire human organ for transfer through 3D bioprinting may still at least be a decade away, according to medical researchers and scientists. Stem cells. Stem cells have amazing regenerative properties already. They can reproduce many different kinds of human tissue. Now, stem cells are being bioprinted in several university research labs, such as the Heriot-Watt University of Edinburgh. Stem cell printing was the precursor to printing other kinds of tissues and could eventually lead to printing cells directly into parts of the body. Skin. Imagine the uses that printing skin grafts could do burn victims, skin cancer patients, and other kinds of afflictions and diseases that affect the epidermis. Medical engineers in Germany have been developing skin cell bioprinting since 2010, and researcher James Yeo from Wake Forest Institute is developing skin graft printing that can be applied directly onto burn victims. Cancer research. In the same way that tissue and types of organ cells are being printed and studied, disease cells and cancer cells are also being bioprinted in order to more effectively and systematically study how tumours grow and develop. Thank you for listening. I will now pass you on to Joe, where he is about to conclude our video. Thank you very much for your time.
Since its beginning in 1983, 3D printing has evolved to become a technology that has huge potential in the field of regenerative medicine. Although the industry has many challenges to overcome before it fulfills its potential, it is still one of the most promising fields of innovation in the regenerative medicine sector.